along this, once we have the separation of chimps and humans, each all evolving on their own paths for 6 or 7 million years. So there are changes in terms of the musculature in our jaw, perhaps. There are changes in the nervous system. We have genes that may be involved in speech, others involved in pathogen resistance and thermoregulation, skin physiology, sperm mobility, other things of this sort, which all are occurring along this particular pathway here. Of course, one of the questions we're going to address is what makes us similar or different to Neanderthals here. But we can see that there are a number of very specific changes or adaptations which have occurred in the human lineage, which distinguish us from chimps. And chimps have their own specific pathway also, which then leads them to look like they are now, going back to this common ancestor here. So there are these kinds of changes that are occurring along our evolutionary lineage. Maybe gene changes in genes, other things. But it's not just that. Given the level of similarity of genomes, it's also the way in which genes are expressed. So we can look at tissue-specific expression. There are various ways in which this is sort of a heat map here, showing you that in the various tissues you can have the certain colors, the darker color in red shows a higher gene expression, the lower color shows it in blue. And the basic point here is to show that the patterns are different for different kinds of tissues when you compare humans and chimps to each other here. So it's not, so we share the remarkable similarity at a genomic level, but how these genes are turned on and off in different kinds of ways across different tissues actually is quite different. And in fact, many genes which show very high signals of selection, you'll see some other charts which will demonstrate this, actually have to do a lot with neurobiology, with energy production, with brain tissues of some kind. Some of these changes actually can be relatively recent in terms of human evolution. In fact, to give you one example of a gene that does this, this is one particular gene which actually is involved in helping cortical synaptic development occur in a developmental sequence. You can see differences in chimps and humans, and you can also see in humans and other primates. And this is the name of the gene here, but basically it shows how, in fact, this timing and the relative density of synaptic plasticity is actually quite different. And this change actually may have occurred once you have the separation of Neanderthals and modern humans. And the basic, just to show you graphically, this is just what's called a sliding window looking across a segment of the genome. The gene in question is right here, and where there's a point of selection, or whether there's a drop-off of selection, you can see it actually is happening here, and so there's a spike in which it seems to be linked to this particular time here, where the divergence time between humans and Neanderthals in this particular chart. So we can use genomic information to try to look at changes in basically overall brain structure and brain activity, neurological function in humans and in chimpanzees, among other hominins. We also have the hominin thing moving then sort of very quickly to another topic of actually considerable change and interest, is basically where we stand within the fossil record. So this is now actually outdated. It changes about every half a minute these days with all these new finds. But the point is that we have a certain kind of architecture here based on different kinds of what's, at least by grades or changes of hominins, and of course the genus Homo is in red here. And part of the real question that's come forward in the last series of finds is actually how do we connect earlier forms of hominins like Australopithecus to the genus Homo. That's going to be the focus then of this next series of discussion points. But even this continues to evolve very quickly based on a new fossil record. So this, again, the question here is, does the demarcation of Homo represent a sea change in terms of behavior and biology? Or are there continuities coming from earlier hominins here which we have to be more cognizant of, and hence maybe greater behavioral repertoire capabilities than we once thought? So one thing that was published back in 2009, the paper published by a series of authors on Ardipithecus, helped in some ways to actually show us where we might have started from going back 4.5, 5 million years ago. We can see then we have a common ancestor here which was not a knuckle walker, was probably more arboreal and so forth, had certain attributes which are common to chimpanzees and ourselves, and helped to establish how, in fact, the two lineages then began to move forward in their own way, moving into much more bipedal locomotion, and in some ways more arboreal, living in forests, and reinforcement of the body for quadrupedal activity. So this was an important change also because some of the theorizing around what this might have meant in terms of social organization, but nevertheless this is an important point of inflection and thinking about where do we see behavior which actually seems to be that of a Homo later on. And then subsequently we found, some of you are familiar with Homo sabida here, these are reconstructions, and its placement here Australopithecus, at least possibly maybe a connection between Australopithecines and the genus Homo. And then we also have Homo naledi, which adds further to this mix, stuff from South Africa. And the reason these are both are important is because they both are occurring in this time period where we're seeing this transition from Australopithecines to Homo, but also because they have mosaic characters, some of which actually look more like Australopithecines and some like Homo. So they imply that this process, actually this transformation from Australopithecines to Homo, is perhaps much more complex, maybe messier, and that may not be exactly a linear process that we try to construct using typical sort of phylogenetic methodology. And it's quite exciting then to try to figure out how these pieces fit together. Other things which have tended to shaken things up a bit is the finding of the Colloquium lithics, if you buy into this, that actually are important because it's earlier than previously thought with Oldowan, which was thought to demarcate the emergence of tool making for some purpose. It extends it back to 3.3 million years ago. And this is into the realm of late Australopithecines and not really the genus Homo. So this complicates our sense about what kind of behavioral repertoire that earlier hominins had relative to the emergence of Homo with Homo erectus. In fact, these kinds of finds are also calling into question when in fact we see modern behavior in the fossil record. So this is, there are different debates about this, some saying it was a late explosion. You remember Richard Klein 50,000 years ago, we had this big explosion of artistic representation across Europe. And this is sort of a counterpoint that was brought forward to say that in fact many of these attributes, which are considered to be modern behaviors, extend back into, at least in terms of the Africa, the Middle Stone Age that you can see here going back into 200,000 or 300,000 years ago. So I'm going to refer to this in the next series of slides because within the last month since I started preparing this talk, there have been a series of papers published that actually shake this whole idea up in some respects, pushing things back in interesting kinds of ways. But at the very least, you can see that in terms of symbolic behavior, material culture, utilization of different sorts of materials, the subsistence extension into fishing and not merely hunting, long-distance trade networks, etc. All this seems to be established within the Middle Stone Age, 100 to 200,000 years before present. Now, one thing that, of course, some of this is subject to judgment, but there are various publications which actually say we need to look a little bit farther back into what behavior of hominins could possibly be. One is this particular idea here, published in 2005, that these scratching on this shell from the famous site of Trinil that Eugene Dubois excavated in the late 19th century, it may show some kind of etchings here, or lines on this shell, maybe for food production, or this is what they suggest as engraving. And if this is correct, then this Trinil site, being 700, 800,000 years before present, maybe a little bit later, suggests that Homo erectus perhaps had some sort of behavioral repertoire which involves symbolic representation, if this is not sharpening a tool of something of this sort. Other things which should shake things up a bit is the purported Neanderthal art that goes back into the 60, 80, 100,000 years before present. Dye here on these shells that seems to be non-arbitrary, which could have been used for different kinds of symbolic behavior, or at least a preparation of art of some kind. And then, of course, this is one of the things that suggested that this is some kind of cave art that actually is present in the Iberian cave, I think, 60 to 80,000 years ago, before modern humans were in Iberia. So this, in fact, is due to the provenience of Neanderthals. Although it shows something of a figure here, it still is not quite the same as the art that we typically associate with modern, at least in the past, associated with this big cultural symbolic revolution in Europe that actually is marked by various kinds of elaborate cave paintings and symbolic representations in terms of carvings, faces, and so forth. But it suggests that in fact, this division between Neanderthals and modern humans, artistically, symbolically, maybe not quite as great as we once thought. In fact, something that is coming out yesterday, was announced yesterday, looking at this transition between these dyed pieces, these refinement of these big hand axes, Acheulean hand axes, moving into more refined stuff in Kenya. Now these are actually dated to around 300,000 years before present. So what this suggests, based on these data, this is published in Science, is that, and I'm going to talk about a couple of these things in a moment, is that earlier some of these shelves and carved ochre and things of this kind are in the hundreds of thousands of years. And if this is correct, at this particular plate, these tools which show some degree of finer manufacturing with different kinds of ochre and black pigment on them, extend that back another 100,000 years or so, back into the realm of archaic humans, or at least the incipients of modern humans at this point. So what I'm doing here is giving you various kinds of yardsticks, temporal yardsticks, or different kinds of measures of modernity, of humanness or hominis in this kind of way, to have you start thinking, trying to put these pieces together for yourself, because in fact, this is happening in real time, and all of us are actually trying to grapple with what this means. Now, something else that's emerged from genomic studies has to do with language. And so some of you are familiar with the FOXP2 gene, the so-called language gene, which actually is important because it's expressed in development. It helps to develop Broca's and Wernicke's area in the left-hand side of the brain, and it's very important in having these functional copies of these genes, because otherwise you have impairment in language comprehension and speech production. So for a long time, it was thought that these unique mutations in this gene, this is just a gene tree showing you various kinds of mutations leading to different members of primate phylogeny. And our branch leads here, and we have two unique changes in this gene which produce the form that we use today and is essential for speech production. The genomic data has showed that in fact, now having Denisovan genomes, Neanderthal genomes, and so forth, that in fact we share the same copy of this form of this gene with these other sorts of hominins. And so the question, of course, now that we're going to begin looking at the issues of admixture between hominins, is what do they say to each other in order for this to actually take place? So, one way I do it, is basically to do it this way with a sort of a Philadelphia touch. But what would have been communicated, based on what we know now about some of the symbolic behavior that hominins are capable of? So I'd also like to look at, again, some of the other newer fossil data that help us to try to understand where modern humans began, at least in the osteological or paleoanthropological sense. So until actually about a month ago, this was what we thought was the distribution of modern human skeletons around the world. The oldest ones are in Kafse in school. You see the oldest actually was Homo kibish, some that before in Herto, some of you may know the Ethiopian fossils. And then you see the distribution across time here in all these sorts of areas. And so at least from the craniological record, this was the story that we had. Well, about a month or so ago, we had data published from Jebel Irhoud of anatomically modern humans about 300,000 years before present. So this is, Morocco is not in the middle of, it's not a typical fossil, where you see fossils in Kenya and Ethiopia and Tanzania. And so this was quite striking because you can see, although there's some robust features in the face, overall brain, the globoid shape is relatively similar, facial flatness. All the hallmarks of modern crania are present in this 300,000-year-old individual. Now, this individual wasn't making these very fine blades and things that we typically associate with, at least with modern humans and the Acheulean, let's say. Using Middle Stone Age tools here that you can see, and then some of these below here actually are level 1. So the repertoire of tools that they had are essentially what you see in other archaic humans, also with Neanderthals. So this is quite remarkable in and of itself because of the location of the fossil and of the time depth and of the tools that are actually present. So what this helps us to understand is that, it makes the complex a story about modern human emergence and dispersal and so forth. It also leads to a very interesting picture as to where we map out where hominins were in Africa. And what possible points of contact intermixture could have taken place prior to the major expansion out of Africa. So here's the Jebel Irhoud up here, the typical sort of fossiliferous areas that have been used to reconstruct that tree you saw before are here. And what's important is that these show you sort of the color code of modern Homo sapiens in this dark orange or burnt umber. The lighter color here is Homo deleti, which is at least hypothesized to be something of an intermediate or leading into the genus Homo, although if this is 200,000 years old and this is 300,000 years old, it can't be a direct ancestor-descendant relationship. And furthermore, there are Heidelbergensis, other types like Cowboy here and shown in gray and some others, actually they're living concurrently with what are considered to be anatomically modern humans. So just like we've seen as you begin looking outside of Africa, there's a greater complexity of hominins here than we once thought, and possibly also the presence here of these types might indicate why we see this kind of genomic signal in modern African individuals that may be similar to what we see in other populations outside of Africa, which is mixture with Neanderthals and mixture with Denisovans. So the story grows more complex. It grows more complex with the finding outside of Africa in the Near East of fossils that are not just 90,000 years, that we've seen in school in Kafse, but also, these types are 100,000 years older than this. So this may represent the initial foray out into Africa or outside of Africa, or just a tentative push, but human dispersals, or modern human dispersal, seems to be much earlier in some sense than we once thought. We also know, as we mentioned before, that we are, we in fact, most of us in this room have some sort of Neanderthal contribution, and you don't have to tell me what that is, but the point is that this is a byproduct of hominid interaction and engagement. And in fact, if we survey the world globally, we can see that this is going from low to high frequency of Neanderthal markers. Or you can see the color coding that many populations outside of Africa have some degree of native or Neanderthal admixture. It's a global phenomenon aside from African populations, although those who came back into Africa bear those. And then Denisovans, which actually the story you remember is a fossil found here in the Denisova cave of Siberia. You can see a whole variety of individuals have some measure of Denisovan admixture, but particularly in this area of the world here. So this all comes with the advent of ancient DNA analysis and genomic comparisons. And so if we take the fossil record we've been talking about, and we take the genomic record that we're beginning to put together, and look at this pattern of admixture, what we see is that the tree of hominid, earlier we didn't really show any kind of lines, but you can see now that we've got the whole picture of the emergence of modern humans and the genetic exchange that takes place between them actually is much more complex than we once thought. It's bushier, maybe more mosaic, reticulated. Modern humans here, but there's admixture everywhere modern humans go with Neanderthals and Denisovans. There's specific admixture between these populations. There are other earlier forms that might have intermixed with Asians, other earlier forms of African, and we still don't know what's going down here with Homo naledi or Australopithecus sebeda and so forth. So this in some sense makes it more confusing and more complex, but in some ways more satisfying in that it is a complex process of population history. And so this is again, what's continued to be refined with these new fossils. So what did we get from the Neanderthals? In some cases, so the question is, what was the byproduct of hybridization? So hybridization can happen in ways that you may have unfertile offspring. We know this is the case in case of donkeys and horses and mules. We can do this. So the question is, if we are admixing, what is the extent of it? What is the sort of the selective or adaptive consequences of this? So what we've learned, among other things, is that many of the genes that are actually the alleles we have that come from Neanderthals affect genes which have these sorts of these health-related kinds of effects. They affect skin thickness to some extent, as you can read for yourself, aspects of physiology, the way in which our blood coagulates, different kinds of neurological behaviors of some kind. So this is actually, as we learn more and more about the genomes, this is some of the people in the room are actually working on these sorts of questions. We can see now that there's this kind of legacy which has longer-term implications for us than we once thought. It's not merely the fact we can detect admixture, it's that there are physical adaptive consequences that actually go along with this. And in fact, what's interesting is that it may not be simply the markers that are in Neanderthals. It could also be in a way in which these particular alleles that we get are actually expressed. So in other words, if you have certain, what integration is admixture, certain traits that are passed down from Neanderthals, that they have to be expressed in a certain kind of way for them to actually create phenotypic variation in humans. Phenotypic variation meaning variation in these kinds of functions or these kinds of attributes. So it may be that it's an allele-specific attribution, and it also may be due, in fact, to the relative, these are sort of what we call quantitative trait loci, the relative transcript, the way in which they're transcribed. How many copies of a protein that are actually transcribed from these genes may actually also have an effect on adaptation of the physiological outcomes for humans. And so even though some of these alleles may not have been ultimately advantageous to us initially, carrying them forward into out of African environments, including thermoregulation, possibly skin thermoregulation and so forth, may actually have been up to humans to adapt to new environments that they had not encountered before. So it's not merely the mutation, it's how the mutation works and how many copies of the mutation may actually be present. Okay, so that's sort of the hominin portion of this, the primate portion. Now I want to shift to a topic that our previous speaker spoke about, which is the microbiome. So most of the cells in your body are actually bacterial. Most of them live in your gut, around your skin, you're in various places. But in fact, we are a composite of many different kinds of organisms. This is why we're called basically, holobionts. We're a combination If you look at the overall host genome, a combination of genes from viruses and from bacteria and so forth that we've co-evolved with. So the microbiome you can separate from the whole genome, and host genome is here, and the environmental genome is other things we come into contact with. But you basically see that in terms of thinking of ourselves as having so many billions of cells, that we actually, there's order of magnitude greater number of bacteria in us, and they play very specific important roles, as you actually heard on the previous presenter about especially nutritional adaptation and things of this sort. So this is a different way of conceiving what a human is in terms of its component parts, and also the parts, bacterial especially, which are essential for us actually doing a number of things well. So for example, this just affects our physiology, it can affect our psychology. I'll give you other examples here. The most significant productive source of serotonin in the body is our microbiome. It's not our nerve cells, for example. And physiologically, so we have to have some exposure to the microbiome in order to have appropriate sensing and toning of our immune system. It's a starting point for our recognition of self and non-self. So these are just some of the basic understandings of what a holobiont is, and much of our genome actually is a legacy of viral incursions into the genome and integration of these genomic segments that actually, some of which have regulatory functions, some of the so-called junk genes. So we have millions of years of accumulation of viruses jumping in and out of our genomes, which we carry with us now as modern humans. We also know the microbiome has, these are specific families or different kinds of bacteria, and we know that even the body itself has a variety of different combinations of bacteria. So the epidermis is different than the vaginal area, the esophagus is different than it might be in the stomach because of acid conditions. And so our body actually has, depending on what we experience with antibacterial soaps or what we eat, we can change the composition through time. We also know that there's an important intersection in terms of microbiome and setting future health and responses to diseases between mother and infant. Passing what a mother experiences may well be passed to the infant through various kinds of contacts. The first contact with the infant, at least with the vaginal birth, sets the initial microbiota and also nursing. And this has some carryover in terms of how the infant begins to develop and the relative way in which the immune system develops. In fact, there's an aging component to this as well. Our microbiome changes through time, from the point at which we're unborn and more or less in a sterile environment in utero, to the point at which we age and we see different kinds of things changing to us as diets change and appetites change and the relative nutritional needs as well as we get into more senior conditions, as it were. So it's a dynamic process that all of us experience in different ways. We also, just to give you some details, other details, that the relative type of microbiome that you have, the things you're exposed to, may also cause certain kinds of problems regarding Crohn's disease or ulcerative colitis. Also what you're exposed to here, and then some of the things that are actually crucial for what the microbiome does for us actually take place in the gut. And what we eat, the antibodies we use, the surgeries we have, our basic genetic makeup to some degree engages with this microbiome and produces a range of responses to it. So for example, this is just the ways in which this shows. So depending on, if you alter your microbiome, you can have inflammation, metabolic disorders, different kinds of immune system deficiencies, relative hygiene or exposure, all in some ways has an effect on your microbiome. And just another slide to show you that here, just with the serotonin, that in fact there's the microbiome. So typically we think of in terms of brain functions, hypothalamus, pituitary, gonadal axis with regard to development, reproductive development, other things of this kind, other kinds of compounds like corticosteroids, which are involved in stress responses. But now we know there's another axis we have to pay attention to because of the effect that in fact the microbiome has vis-a-vis the intestine through the brain, relative response to infectious agents, immune tolerance to foods of different kinds, and then also the relative solute concentrations that we maintain in our bodies. So the microbiome, the notion of human as a holobiont, is now a new way of conceptualizing what is human that extends beyond literally the human tissue and human cells to the organismic that we've been evolving with for a long time, which can be altered in real time, even after eating foods. It can shift very subtly in terms of the overall microbiome composition that we have. But evolutionarily speaking, there's been a coevolution, and it probably has changed, as actually the previous speaker spoke about, going from hunter-gatherers to farmers to industrial societies and so forth. And these have real-world health implications. So another thing that now has come back onto the map, shifting from the microbiome, is this notion of theory of acquired characteristics, or what we might call epigenetics. So some of you may recall that Darwin's theory of natural selection through descent by modification was a view that was in contrast to many others who were working at the same time or slightly before, including someone like Lamarck, who had a different way of thinking about this. And the debates about human evolution, or evolution in general, was whether, in part, how if you had this kind of phyletic transformation that changed more or less unchecked through time, or whether you had sort of branching and stopping reticulate kind of process that led to different kind of speciation. And so the reason that his ideas are being revisited, at least in part, has to do with this notion of soft inheritance of acquired characteristics that actually are passed down transgenerationally. So the typical, I'll give you the classic example that's used for him in this case, but the point is that his idea about this soft inheritance was one that he actually drew upon from other researchers, and it was part of what he was trying to talk about with regard to this notion of transformism, where all lineages continue are basically an antigenic sort of pathway. So in other words, once you have hominins, they never really stop being the same lineage. No speciation, nothing of this kind. It's just one big lineage which continues for millions of years, as opposed to a tree-like structure of branching and reticulation and so forth, where some lineages go extinct and others persist through time. So this is why it contrasts, this idea of contrast with the notion of gradualism, or the stepwise process that Darwin put forward, and why, in fact, he, to some extent, rejected Lamarckian ideas. But this, right now, in terms of what gets passed down through generations, has come back to the fore because of a growing understanding of epigenetics. So this is a model which I'm sure most of you are familiar with, about the idea that through time, through use, the neck of the giraffe grows because as the trees change, or that there's better nutrition gained from eating, that there's selection on taller necks because the giraffe is stretching the neck to reach the leaves, and this stretching, the physical consequences of this in the parental generation, then passed on to the next generation here. Now we know that's not exactly the case from an evolutionary standpoint, but this is the model that's been put forward. What we do know now is that the soft inheritance that's modeled by this in a general sense actually works at the level of the genome. And the reason this is important is that it's not, in this case, just the genes you have. It's how the genes are turned on and off. So there's a very intimate type of, there's a whole series of different enzymes and marks and methylation and so forth, processes, which regulate how tightly wound or how loosely wound your DNA is, how open it is, so that other polymerases and such can do things like make copies of genes and so forth, or if it's restricted such that the genes are actually effectively unavailable to those particular enzymes and simply are silenced. And so as we learn about the different kinds of marks and the way the histones are wound and spooled back together, we now begin to understand how the genome functions and operates. And much of what we know now with regard to health and disease and other risk factors may not be simply on the genes you have, but how they actually function, how they're active or activated. So for example, we know this is true in transgeneration. There are genes that are marked coming from both parents in this case, but we know that the marking takes place in the gametes, changes here, you have demethylation to produce the zygote, and then through time in development you get a series of marks, largely from the uterine environment, which actually marks genes that may involve with metabolism, that may give signals about the relative nutritional environment outside, the exposures the mother might have, and so forth. And then once born, you continue to develop these through time, such that you wind up having, it can affect the way that you age, your relative risk for cancers and dietary disorders and so forth. So the point is that this actually starts from the point of conception. Actually, it's probably a transgenerational effect going back at least to where, because a mother in a pregnancy represents 3 generations. The mother, the fetus, and then the germ cells in the fetus itself. So you see that this process is quite dynamic here in terms of the marking along the developmental pathway, and then the experiential component of this that happens once you're in adulthood. And this is why, in some respects, you can have persons who are identical twins who can wind up having different kinds of lifestyles and risk factors and exposure to cancers and things of this kind. Same genome, different epigenome in this case. So what this raises then, with this basic knowledge of epigenetics, without going into all the molecular details, is that this raises some interesting questions about what is nurture and what is nature. How do we understand this to actually happen within humans now, especially with this understanding of epigenetics? So we know now that, in fact, various kinds of things can impinge upon the epigenome and modulate various kinds of experiences here. This next slide also just shows you that in fact, the embryonic development and lactation can affect the phenotypes, the placenta. And depending on the environment in which individuals find themselves, this could affect the developmental course that individuals go through. So if they have a normal development in a safe environment, then you have normal aging, normal development, and so forth. If you have metabolic disruption marked by epigenetics and so forth, or exposed to harmful environments, toxins, pollutants, whatever this is, this actually may affect acceleration. It can also cause problems in terms of diet and health in general. So for an example here is with looking at basically at the testes production. So depending on the environment that the person experiences, the male experiences, smoking, works in a factory with pollutants, whatever it might be, this affects sperm production and marks these sperms. This changes in some ways how sperm are produced, especially with dietary considerations. And then this particular outcome, then when you have fertilized eggs and when the father has children, his experiences and exposures can be transmitted to the next generation through sperm cells. We're now learning, we can see this in mice, and now more is being done looking at various human populations as well. But it's not just reproduction, it's not just diet, it can also be stress. So in fact, this notion of allostasis or buffering of different kinds of stresses, the type of epigenetic signatures you have, what you experience, both in terms of what you experience here, but also what you're born with to some extent, the relative degree of stress, actually can influence the way you respond to different stressors in different ways. And if it's a serious enough kind of stress load or allostatic load, this can in fact cause various kinds of endocrine disruptors, can cause problems with health, stroke, atherosclerosis, and so forth. So early life adversities, if you're poor, if you're impoverished, undernourished, this can lead to the stresses here, environmental pollutants. This can have an effect on your epigenetic signature, which then intersects with the environment to produce different kinds of phenotypes. So there's a lot of details in here. I'm trying to give some basic gloss on these points about what we're learning about epigenetics, but I think you get the idea that the nature and nurture here are not so finely divided. This, in some sense, that there's a division. You can certainly define them in some sense, but it's a process of engagement and interaction between these features which give rise to various phenotypes, the behavior and biology of humans. In fact, as we begin to learn more about things like circadian rhythms, autophagy, which is the process of recycling cellular material, epigenetics, which actually regulate energy function and so forth, and calorie restriction with regard to diet, we even now have greater insights into how we can actually modify longevity, where we might be able to take action to actually extend lifespan, and also where we might be able to treat or reverse different kinds of pathologies which affect neurological tissue, which give rise to epidemic diseases or increasingly huge diseases such as Parkinson's and Alzheimer's. So in other words, this melatonin is one of the things that can be used to modulate circadian rhythms and has an effect on these other things. But the main point is that we're beginning to identify, both through epigenetics and also through understanding of the molecular architecture of the body and aging and so forth, which compounds actually affect this energy metabolism, to some extent mediated through the mitochondria, such that we can then intercede in different ways and possibly extend lifespan of humans. And this is going to the point where it's now pharmacologically being advertised in this kind of way. There are different things we can use to blood-borne, this metacurin, for example. There are other things which can involve stem cell therapies to extend lifespan of cells. How you get rid of senescent cells, so you only have healthy cells. Different kinds of boosting your mitochondrial function to generate more energy, etc. So all these kinds of things are sort of reverse genetics in some sense. Once we know about these processes, also how you modulate epigenetics in some kind of ways, you may be able to actually extend lifespan or treat different kinds of diseases because you know that basically we begin senescing when we're born. We continue to grow and develop. But these sorts of treatments, using this understanding of human biology and biochemistry and physiology, may allow us to actually extend things, our lives in different directions and deal with different diseases which have been quite significant for a long time. So what these sorts of things, this nurture-nature argument in some sense, or this kind of presentation I'm getting at to a larger degree, is getting at this notion of niche construction. So niche construction, I think some of you know this concept fairly well, but basically, we make environments which affect ourselves. Beavers make dams. They dam the stream. It creates a pool where there's food. They have a place to live and reproduce, and basically it suits themselves. Ants create colonies, the various examples. We're certainly, in terms of niche construction, we're pretty good at that. But the idea, at least in terms of modeling them, because I'm talking now about, with all the epigenetics and so forth, it's sort of a modern take on this kind of concept. But the reality is that the various kinds of models talk about how this works in long time. So what this is simply suggesting here is that there's an interplay between natural selection and the genetic inheritance and ecological inheritance, including culture, that leads to this back and forth, and selection acts on humans who are actually being influenced by the environment in which they actually are living. So if we begin agriculture and start shifting food sources, this has a big impact on what we're selected to be able to eat or be able to consume. And this has been seen through a number of comparative genetic studies on a number of genes, and this is probably an 8-year-old slide. All sorts of genes here actually have some impact on our physiology, whether it's skeletal development, energy metabolism, skin pigmentation, thermoregulation, whatever this happens to be. And there are various kinds of links to cultural practices that we actually know are probably linked to the increased frequency of these genes being acted upon by selection. Malaria is a big one also. The classic case of this, basically what you've heard last week actually, is lactose persistence. Wherever you have dairying in some significant way with cattle and certain animals, you have high frequency of lactase persistence allele. And this only exists in populations that actually are eating and consuming milk. So this notion of niche construction is quite important in this regard. Now there's a recent study which is, I'm still digesting some of the details here, but I thought I'd throw it out here just for general consideration, because this talks about the role of a lot of the discussion about certain kinds of attributes or intelligence and so forth that tend to be, at least in the past, that lean towards a more hereditary explanation. You have certain genes, so you're more intelligent in some ways. This is a model which is trying to get this in a sort of a different kind of way, this less of hereditary and heritability study, to talk about the interplay between genetics of parents and the genetics of offspring, and what happens in terms of this kind of, what is genetic nurturing, or what is the effect of the environment, basically genetics. And so this is a complicated model which is trying to show you how the parents can contribute alleles from their genetic nurturing, the genetic tendency to be able to do something. And the phenotype of the offspring, and then at least in the genetic pathway here. But the important point is that these particular effects don't happen unless you have the right cultural environment, the right social environment, at least in this basic model that's here. And this is simply showing it how it might work in a transgenerational sense, with various kinds of grandparents or sibs or something of this sort. But the takeaway message from this study, and again it's published very recently, and it's something just to sort of throw out here, gets us away from, at least with regard to intelligence, all these hereditary and strictly hereditary notions, is that this genetic nurturing, even if the alleles are not carried by the gene, the genetic makeup, the variants maybe of the parents can affect the fitness of their kids. Even the kids don't have that particular allele, because you can actually show that the variation that they give to the kids and those without is actually affected by the environment in which they live. Okay. So the only way in which these alleles have effects is through the environmental interaction, gene-environment interaction. So this allows us maybe to escape some of these sorts of these gross generalizations about who has the right stuff to become an astronaut or become an engineer and so forth. It's a byproduct, our successes, maybe this is common sense understanding, but at least this is theorizing how in fact you can conceptualize this process of genetic innate abilities to some extent, if you will, but the environments in which these kinds of talents or abilities and so on begin to be flourished, can be realized. So let me come to a couple of then points which gives into biotechnology, and I'll move this the next couple of steps relatively quickly. But I wanted, in this section, to think about not merely where we've been and where we come from and sort of what we know now, this transformation, especially in the Neolithic, but also talk about what we're doing now and how, in fact, we're actually actively acting in some ways to be much more direct in modifying our future directions in some sense. And a lot of this comes through biotechnology. The earliest part of this has to come through in vitro fertilization, where you're basically taking eggs from an ova from a woman, fertilizing them in vitro, the test tube babies, reimplanting the embryos, and then gestating the fetus to term. And so this is relatively common. It's done with surrogates as well as with biological mothers. But this was the first step towards possibly modifying how we do things with regard to human reproduction. What we can also do now actually is create germ cells through stem cells without actually taking ova or sperm from the germ cells to be able to produce an offspring. So here is just, you can look at the steps that are here, but you can actually produce fertile offspring. This is using mouse experiments where you're actually creating germ cells in vitro, not taking them from the individuals themselves. So this raises the question, what can you possibly do in the sense of actually inducing stem cells to produce germ cells with regard to the genetic makeup of these particular cells that produce new offspring. Another thing that's happening that has to do with modulation of genomic makeup, but also with disease, has to do with creating basically sort of called a triparental offspring. And the basic idea here, as you can read this, is that you're basically, because mitochondrial mutations cause huge numbers of diseases, many of which are pediatric at their basis, and which have devastating consequences for young people who don't live to adulthood, as well as in terms of long-term senescent types of diseases. If you can intervene, especially at the earlier stages with these pediatric diseases, by basically sucking out all the mitochondria, leaving only the nuclear DNA here, putting in a healthy mitochondria into this egg, and then producing this reconstructed egg with a healthy mitochondria and a normal genome, you can then implant this into a woman using the same in vitro techniques, and then basically create offspring or have offspring born who don't have these kinds of energy deficiency that actually occur because of mitochondrial dysfunction. This is already being done. It's done in China. It's being looked at in terms of gene therapy here. And the main reason that this is important is because mitochondria are essentially for your energy. And if you have energy deficiencies in your nerve cells and muscle cells and so forth, you're not healthy. And furthermore, what we know about this is that there's sort of what's called a threshold effect. So people can have normal and unhealthy mitochondria in their cells, and the relative ratio of healthy to mutated forms basically produces a certain kind of a phenotype. The more that ratio is skewed to the mutated form, the unhealthier that you happen to get. So if you cross a certain kind of threshold, you more or less are going to have to expect to have some serious metabolic and physiological problems of various kinds. And the reason that this also is crucial because these kinds of mutations, what did I do here? Sorry, are actually systemic. They clearly identify areas which are highly oxidative in nature, but in fact they can have multiple different kinds of effects. And also because these mutations, depending on if you could remember back when you were an 8-cell embryo, okay, you start to have various kinds of tissue differentiation into major tissue groups. And so depending on how the mitochondria are sorted, the healthy and the mutated are sorted into different parts of your body, you can have the same mutation can cause different kinds of phenotypes. And the relative ratio then is going to tell you whether you basically have neurological problems or heart problems or some combination of the two. So that's why they're trying, if you can intervene at this level here, and especially in terms of if you're using the ova of a woman who has the mitochondrial mutation, she may have healthy offspring once you do this kind of parental type of a mix. But this in itself, that this mitochondria is coming from some other person or some other woman. So the offspring are not entirely genetically that of the parents, but nevertheless are healthy. Again, giving rise to the question about what does it mean to be human, especially if you can use CRISPR to sort of doctor up or take care of mutations that you also have in your genome at a nuclear level. So the basic idea is that you can use tools that we know that are actually in yeast and other species to go back in and basically take a donor DNA and repair mutations which actually cause various kinds of problems. The reason this is green is just to show you that these actually are sort of transgenic animals, but you could actually edit the genome. Taking out mutations which might cause diseases of some kind, may cause neurological disorders later in life, things of this kind. Now, some of these are not quite at the level of clinical trials and such, and there's certainly a question about what this means and ethically and so forth. But you know, where is this going to lead if we have the ability to actually modify our genomes in this kind of way, not just doing it the old-fashioned way of reproducing, but in fact intervening in it much more substantially. Now, the way the response to this has been, at least if you look at some various kinds of information, is that in terms of making the baby more intelligent, there are certain responses to this, but if it's basically to get rid of these diseases, it seems that at least some number, at least half the people who are surveyed are willing to go that route. But that has longer term implications about what is our genome going to look like and how many generations from now if we have the ability to make these kinds of changes. And not just this, we also have, cloning. Some of you may remember Dolly the sheep, some of you may not, right? But a case that's been recently in the news, right? She cloned her dogs because she just couldn't be without them, right? But actually about 6 months ago, maybe less, macaques were cloned. Now macaques are not too far away from us on the evolutionary tree. And we have natural examples of clones, in fact, in terms of triplets, or that basically are the same. But the question is, what are we? This is also the stuff of science fiction movies, of course. But the question is, is this something we want to get into, or what does this mean in terms of extending lifespan beyond, beyond a normal period of time? Furthermore, if you like science fiction and these sorts of things, there's the whole question about, in terms of human cloning, what is the essential part of being a human? Now, I don't know if any of you know about this film, but the basic idea is that the bodies in this movie are called sleeves, and basically you have a hard drive type thing that's stuck in the back of your neck, and that contains your essence and your neurological profile and all your history and so forth. So you can be, you as this entity, this virtual entity, can be inserted into any body, whether it's a male or female, old, young, kid, guy with one leg, whatever it might be. And that's your sleeve, that's who you are. You don't change, but your phenotype has changed. So this leads to the question about what is the essence of humanity? Is it a disembodied form of human in this sense, or is there something really important about the essential linkage between the physical body and the cognitive self? This, of course, is also the basis of other machines here, like these movies Ex Machina. But in reality, in some regards, we're not so far away from this. We're creating prosthetic limbs. My mother is still living because she has a heart valve transplanted, an aortic heart valve, artificial skin. We're growing various kinds of other things, brain chips. So the question then is not only is whether we can extend life and so forth, but if we're going to create all these artificial organs, where do we stop being human, or do we, if we add these sorts of enhancements to ourselves? There are examples already, especially from veterans of Afghanistan, being able to use these arms. And in this particular case, this individual here is trying to actually move his arm to the connection neurological to be able to think himself, rather than just have to do it as more or less a mechanical kind of way. And furthermore, we're beginning to use our own cells to create banks of stem cells to be able to produce new organs of different kinds, or different kinds of tissues that can be used for transplants of some kind, or growing new organs in case of kidney failure. Example of this already exists with regard to replacing damaged windpipe. This is just one example of sort of tuberculosis. The area is damaged in the trachea that's here. The individual stem cells are grown to produce tissue. The tissue that actually will be recognized itself is wrapped around a specific tube that's inserted surgically into here, that is covered with cellular tissue of the individual so that he won't reject it because of immune response to something foreign. So the question then the last couple points I want to ask here is that are we approaching this kind of a cyborg reality, and where do we, what means human actually, where do we place ourselves here with regard to being human, certainly from an anthropological standpoint? The final thing I want to ask is, where are we going with this? And you can read a lot of literatures of this, transhuman literature, other things, but at least some suggestions there that what we might expect over some number of generations in the future is that, if we may become more similar to each other, live longer, changes in our basically immune function through exposure to different kinds. We actually may be demographically all heading to a much longer reproductive lifespan, or at least later aging child reproduction. Some concern about the degree to which we communicate as mediated through electronic communications and such, and also this whole question about this interface with computers and actually planting chips and brains and things of this kind. So this is what I would like to leave you here in thinking about, again, what it means to be human in the post-genomic era. There's some very interesting trends we see here, but I hope this gives you at least a sense for at least how this notion of human and hominin has been changed, even within a matter of a few hours or days based on new literature. And I'd be happy to hear your thoughts about these particular ideas. Thank you. Claire?
I'd just like to hear some of your thoughts on so I watched a little bit of the Altered Carbon, so I'm gonna put it in that context, but in that show, there were sleeves, but not everybody had access to them. And I think that would be actual future for all of this technology and the present. What are your thoughts on what is the future of inequality in the context of all this technology and information?
Well, certainly if you read the literature on these kinds of projections in the future, the issue that always comes up is an unequal access to these sorts of medical resources. We know what happens now in this country and other places, and who has the health insurance can pay for these sorts of things may well dictate who at least are the first individuals to be able to take advantage of these kinds of medical interventions of some kind. The CRISPR techniques and the mitochondrial stuff, that's very expensive because in some ways it's still somewhat experimental, but it's still being done, and so the costs are actually extraordinary. But that, I think, is always going to be a part of this story, and I'm not sure how we can, in some ways avoid that unless we democratize medicine or access in some ways to all people, especially those who have significant health problems.
Probably an unanswerable question, but does this increase or decrease world population?
Well, this is the question. Was it, if we live longer, this is the demographic question. We have an aging population now. We're changing the way in which we can reproduce. If we live longer and we have longer reproductive period of time, we may not solve that big population crunch now, but perhaps through time, longer term, there may be a shift demographically, so the population possibly would go down. But these two things, especially if we're able to produce more and more food and things of this kind, world population growth may not be solved overnight. It may take some time before you have any kind of stabilization, especially if we are lived, So we've been able to live decades longer merely by removing diseases at earlier ages of life and a lot of other diseases in general, and also by having better food. We live now 70, 80 years old, and some of these projections are we could live to be a century without a lot of problems if we can tweak all the different kinds of points at which we mitigate senescence and actually extend that time before we actually sort of reach the crunch time as of physiological integrity. So, but if this also means the question would be is if you stretch out this lifespan, does that also stretch out aspects of your reproductive life? Can you have kids successfully as a woman at 60, like you see in the National Enquirer, or is it what are we stretching in terms of human lifespan in this regard? Is it just a life post-reproductively or not? That's one question which actually is a significant one with respect to your question.
Did you address sperm counts declining as being an epigenetic effect?
I haven't spoken directly to that, but
that would be part of that.
Right. Through stress or environmental exposures and other things, right? Sperm counts are lowering, male fertility is diminishing, so this right.
So I say it's a wash, probably.
Thanks, Pat. There's a lot happening, right? Particularly in the last decade or so, in the way that you describe it. Why don't you say a little bit about why or how attention to microbiome or in relationship to soft inheritance, why the development is happening now, when in some ways we did know that we had got bacteria for a long time, right? So what is happening that makes us attentive to these sensitive developments in human biology as well as anthropology now?
There are probably a couple of things that have helped. One, with regard to epigenetics, it's probably the ones that have biological in nature probably have to do with the actually having the tools to interrogate questions concerning epigenetics and genomic evolution and so on in ways we couldn't before, because we didn't have the data, the computational ability to understand the information. The second thing is probably the realization that that for some people, let's say with the microbiome, yes, we've had it for a long time, but a lot of the work understanding its role in diet probably came from animal species, agricultural work and so forth. And so just a matter of realizing that the humans are actually not discontinuous from that part of the natural world, and that we actually ought to look at the same questions there. So I think it's probably a couple of those sorts of reasons. The epigenome in particular is a complicated thing because it's, I mean, if you try to do DNA sequencing of a microbiome, you have an order of magnitude large. If you take my microbiome, you sequence it, or you try to understand my genetic makeup, a small proportion of the overall genome is actually mine. The rest is bacteria. And then you've got to sort out which bacteria are there through a variety of different kinds of computational techniques as well. So even understanding that part actually has come a long way with genomic sequencing. Although the recognition of diverse species being present was always there because you could culture bacteria, you could actually see them morphologically and behaviorally to see what they were like. So it's probably a combination of the recognition that there's reason to look at it with regard to humans, but also the means of actually interrogating questions concerning health and disease vis-a-vis these particular systems of marking or systems of nutritional biology, let's say. Sure.
Go first.
Just looking at your examples of Neanderthal introgression in humans, I was very curious that all these examples seem to be negative, the deleterious variants sometimes. And I was wondering if that is a bias of the method, just because it's easier to identify deleterious variants, or if that is sort of universal as an incompatibility in the two genomes. So, what's your take?
Well, I think there was a paper published maybe 2 or 3 years ago out of one of the researchers working in Svante Pääbo's group, who has been looking at that for a while, and the question that she and her group were trying to assess is basically the consequences of hybridization, because hybrid infertility is one that can actually happen when you have genomically close species who just aren't close enough to actually produce viable offspring. And so I think some of her work showed that there's especially on the X or the Y chromosome. The X chromosome, I think there are certain genes there which are underrepresented in terms of Neanderthal versus modern human variants, which suggests some kind of problem with fertility, maybe vis-a-vis males, that you would not expect if you had viable offsprings who are close enough to be, like some species of snakes or some species of birds who can hybridize, or baboons, for example, who can do that. So I think there's still some sense that that's part of the story here, but I'm not sure how far that's going to work out exactly. In terms of deleterious alleles, I think these I think the way that I showed you are actually they're risk factors for certain kinds of diseases, but the one that causes the keratinitis actually, at least in a mutated form, may actually also be protective against in terms of cold temperature and other things of this sort. So perhaps those are showing you the deficiency of the problems that can be caused by those alleles. But some of them actually are adaptive, as you know, for human populations. But of course, this is the genomic scan. We don't have epigenomics from Neanderthals. We don't have other things. So it's a first approach to trying to get at this kind of question about the implications of hybridization.
I have kind of a naive question about that. I know that I don't know enough to ask a good question, so I'm going to ask a naive question. Looking at your kind of animations of the two the contrasting of your gene therapy examples with your genetic nurturance examples, it just seems to me that the antithesis of nurturing would be unwinding the genome and ripping out parts you didn't like and cramming in some new part. How do we understand the role of epigenetics in these new cells?
That's actually a very interesting question. That's probably why some of these things have not gone so far into clinical trials as of yet, because that's something that I don't know that they were initially thinking about that to begin with, but now having this greater understanding of epigenetics, especially in utero, they're somewhat more cautious in moving forward because of that kind of reason. But in principle, the idea is that you can, in some ways, fine-tune the genome to remove deleterious alleles and then supplement them through either through editing or replacement with other forms which are normal, if you will, or at least are non-deleterious. But that question, I think, has not been resolved as yet. That's still, I mean, if you read the literature on this, there are recent papers published about how we do this with respect to the epigenome, with potential influence of the epigenome. So I think it's still something of an open question. Yeah.
So you spoke to the issue of sort of natural selection and evolution as a model, but I was not yet clear as to what you're really proposing there. in terms of the classic Darwinian model extended through genetics, that you get mutation, selection, passage of genetic material on there. You've mentioned niche construction theory and epigenetics to go along with it, but basically, what's happened to evolutionary process? First of all, in the human species, are we even evolving? I mean, that's been a question lots of people have been asking.
That's right.
Are we even evolving in the human species? But you're suggesting even in the animal world with the processes that were, like with the giraffe, that we've taken to be classic kind of examples of Darwinian evolution may not be actually accurate ones. So what do you think, what can you tell us about what the state of evolutionary theory is at this moment?
So I think for a long time there was a battle between evolutionists and mutationists who thought that their difference debates early on in the field were about mode and mechanism of change. And the fight between Darwin and Lamarck was about the notion of heritable material with variants being transmitted to each generation to produce new forms on which selection could act. And Lamarckian theory was problematic because if you blend or you just simply acquire things through experience, that may not produce the same kind of selective material that needs to be acted upon by selection. So what's changing about evolutionary theory in some ways is that those two perspectives, that both dealing with transgenerational changes, are now seeing the genome in some ways as the raw material of evolution. But also one that actually is modified through soft inheritance epigenetically, transgenerationally, in ways we didn't actually account for in the past. So I don't know if it's an exact merger, but it's bringing these ideas together. So that's part of the answer to your question. And so what I was trying to present here is sort of evolution, or ways in which selection and evolution can work at multiple levels, from the very molecular to basically the cultural. So. You can look at somebody who's lactase persistent and study the history of the adaptation based on the markers that person has. You can trace the ages of mutation, the lineage it comes from, and so forth, and the physiological outcomes that person can drink raw milk and not be flatulent and so forth as an adult. And so, excuse me. But if you want to also look at about how the range of the expression of that gene may work, it could also be their epigenetic influences on the relative ability to digest milk. That person may have other sorts of genetic alleles that may influence the way in which that person can metabolize all those sorts of things. The microbiome has been shaped in a way to be able to metabolize lactose, sugars, and so forth, and you can see that in the makeup of that individual. So I'm trying to make it a more complex story, basically. If you look at the macro level, it's a story about humans engaging with the environment and making changes which influence them, have long-term downstream effects, which we can actually see through interrogating the genome. But we can also go down to the level of the molecular, even of just transgenerational changes. You see, it's not just a simple matter of what you get from your parents. It's a combination of epigenetic, genetic, and other sorts of influences that make you both connected to your parents and also as a distinctive individual. So depending on the time and the scale that you're looking at change, you may have a different kind of explanation for how evolution actually is working. So I think the general view is that humans are still evolving. Many work, especially for looking at long-term projects, like what's the one up in New York, long-term heart study. Basically, you can see that in fact, over several generations, women who live in upstate New York are actually changing in terms of their overall stature, their overall relative cholesterol profiles, and relative actually resistance to heart disease compared to their grandmothers and great-grandmothers. And that change in terms of allele frequencies and those sorts of physiological phenotypes, actually, you can see them happening transgenerationally, but it's subtle. So we have that ability to see things transgenerationally in terms of those long-term population cohort studies. But I hope you get the idea that depending on what question you're asking about the nature of evolution, you can look at how humans have evolved from primates, comparing genomes that show us 6 or 7 million years of evolution going on. Natural selection has not been replaced. It's not that it has gone away. It's just that what the factors that actually are acting as selective agents are environmental, which are both biological and cultural. So that's the interesting part of this, is to recognize that we, in some sense, are actually working in ways that actually have feedback on ourselves, and have probably been doing it for a very long time. It's just the recognition of this and trying to model and explain it for both the gross morphological and behavioral level down to the molecular level.
Would you care to speculate a little bit on how this might change, along the lines of what Greg was asking, how this might change our understanding of inheritance and heritability?
Certainly it changes our understanding of inheritance. Now, as for heritability, the proportion of variation in a trait that's due to cultural or genetic factors, that becomes a more complicated equation. If any of you study heritability, you know that there's a certain formula for that. But if you have to add in not just simply a person's genome, but a microbiome and other sorts of things, then that becomes a more complex term to be able to calculate. But is in some ways this would reflect what we now are seeing with this new knowledge of genomic variation in humans and the factors that are heritable in this sense. But it does, it makes it more complex to define and to model. That's why the models I'm showing you in some cases are relatively tentative. They're just an attempt to try to talk about how this and get away from just very gross hereditary sorts of explanations which don't get us anywhere in a whole variety of levels.
I'll just throw one out. A brave new future. Computationally, are we going to ever arrive at a point where we know what is the epigenetic effect and what is the alleles acting to create a situation where there's, health or illness or whatever kind of effect you may be considering?
Well, I think we now can sequence human genomes for some number of thousands of dollars, and in some ways know most every base pair that we would possess as our genome. So I think that's less and less of an issue. The question, of course, is how do we actually characterize the epigenome, since that actually varies from tissue to tissue and so forth. So we might be able to do something along those lines, looking at adults or even of children of different ages, but how you would do that in utero might be a little more complicated, but that would be a more complicated undertaking, although it depends upon how you define risk with the alleles that you bear. And then, of course, epigenome is a byproduct of exposure to different kinds, which are environmental. So the same person, the epigenome of twins could be very well different. So it's always I would say, probably going to be an approximation, at least at this point, even with the computational tools that we have right now. That's, I guess, the answer I'd give it. Amber?
Hi, Tad, thank you so much. So I'm thinking about so many of these categories that are proposed here. So many of them are responding to something, right? They're responding to so many of the ills of our current moment. And I'm wondering, hypothetically speaking, if we were to achieve all of these things, what might be the challenges of this generation of people? What might be some of the things that continue to persist and plague a society of people who are living longer, resist complex diseases, resist democratic change, maybe don't have as much phenotypic difference, or maybe class and racial difference becomes kind of equalized. What might you imagine might be some of the things that genetic playing around doesn't fix, and that this group of individuals would have to face?
Well, longevity, aging has its own challenges, as we all know. It just extends it into the future to some degree. But what it means, there's basically a financial cost of, already there's a huge burden for countries and nations around the world because of aging population and the fact that, many people are living, 30, 40, 50 years beyond primary productive years. So there's the cost, the financial cost of living longer, which is going to have to be borne out by society, or at least by the populations in which you have many centenarians, let's say, hypothetically. Phenotypic similarity, this is simply an observation that maybe due to the fact that the world is increasingly globalized and it's possible for people to marry persons who they might not have encountered 2 or 3 generations ago, and especially with greater urbanization nowadays, it's possible to, you know, to meet people of different backgrounds and races and create families. So the idea is that that form of contact, so these are sort of long-term trends. So it's not going to happen instantaneously, but that might mean that there would be, you know, hypothetically there might be less difference, you know, less concern about difference per se, and maybe the world would actually be, you know, be changing in some ways to look like it was before you had the advent of really profoundly different phenotypes, like very, very white skin color, which probably emerged 5, 8,000, 10,000 years ago. So that's maybe part of what you might see in those populations. The disease part of the story, this is just hypothetical, but if we can resist the body, you know, complex disease, or I mean heart disease, other things of this sort, hypertension and so forth, this can be mediated by some kind of ways. But if we, you know, we're susceptible to, if we have, you know, if you get more genetically homogenized, let's say, hypothetically, then we're more vulnerable to certain kinds of diseases which can affect a larger range of people. And since we already know, like flu and other things that some of you have experienced as a global phenomenon, and certain forms can kill a lot of people, or at least leave a number of people vulnerable because of immunological vulnerabilities. If you have the greater homogeneity, this might mean that epidemic diseases could actually have a bigger impact on populations because of the relative inability to resist. So the demography, I mean, this has to do with how fast the population reproduce, and so you know, family structure. This may have some implications for how you have social organization in different kinds of ways. This is something that people have commented on because everyone's looking at the phone and putting on virtual reality goggles, and in some ways preferring this, you know, preferring computers to social relationships. And so, you know, I don't know if this is, I mean, these are speculative in some sense, but the question this raises is how do we engage with each other when in fact we are having more and more social interaction mediated by some kind of electronic medium through which that action takes place. We're a very social species, and maybe all we're going to do is just adapt these sorts of resources to different kinds of social engagements, interactions of some kind. But the question is this going to create some kind of alienating effect for humans such that we actually don't react to each other the same way that we do now through more kinds of face-to-face interactions. So at least those would be some ways of responding to this. I mean, this is not meant to be any kind of panacea, as so much as just things that looking at what's happening now, what might possibly happen in the future, and at least some explanations for why that might be the case. I mean, these are coming from models of things that happened within 10 generations, some in the next 1,000 years. So if you're looking at evolutionary trends, you have to take that kind of